To be able to introduce Charles Arntzen tonight. He's the Florence Eli Nelson Presidential Endowed Chair at ASU. He's a professor in the School of Life Sciences and at the Biodesign Institute. And in fact, he was the founding director of the Biodesign Institute. What was that, 2000?
2001.
2001. Dr. Arntzen has made some really terrific discoveries in his career, and he's received numerous awards. I'm not going to begin to go through the list. Perhaps the most prestigious one you know of, Dr. Arntzen was elected to the United States National Academy of Sciences, and he did this, I didn't realize it, at a very young age, 13 years after being a postdoc, which is practically being a grad student. He was elected into the National Academy. That's pretty impressive. So there were some important discoveries that went on there. I think maybe the discovery of aquaporins might have been a few years faster, but I don't know. That's fast. So as you probably know, that's why you're here. Dr. Arntzen's lab provided a lot of the foundational work for this experimental treatment. It's now been used on 7 up to 7 U.S. aid workers that had contracted Ebola. So it's called ZMAP. He's going to receive an award next week for this work, among other things. Tonight you get to hear about the science behind his work. I just get such a kick about thinking about this career that Dr. Arntzen has had. I mean, he started with a really basic interest in cell biology, plant molecular genetics, and protein engineering, and ended up working all these different developing countries around the world on food safety and health and food productivity issues. Wow. And I want you to think about this while you're thinking about how am I going to make this path to success? How am I going to make a difference? And you don't know. You don't know how you're going to do it. I want to ask Dr. Arntzen if he knew as an undergraduate that you were going to end up treating Ebola, or have even the slightest idea that this was possible. What you're here for as an undergraduate is to get the tools that allow you to think creatively and to think deeply about problems, and then apply them into new contexts that we can't begin to imagine. Right? So you're not here to become a specialist now. You're here to develop your minds, and I think you get to see a great example of that tonight. So from basic cell biology, plant genetics, into industry, to biodesign, and to Ebola. Hey, pleasure.
Thanks, Miles. You asked what I was thinking about when I was an undergraduate. I was not thinking about where I was going. Halfway through my undergraduate training, I got kicked out of the university because my grades were terrible. I was out for a year. And I don't regret a moment of it. I loved my undergraduate years. I had just one hell of a good time. But fortunately, halfway through, I met my wife, and she sort of put some vision, or put some blinders on me, so I began to focus and subsequently enjoyed studies as much as I previously enjoyed all those other good things you do in your undergraduates. I'm going to talk to you today about a variety of things in science, but all around this theme of what is it that has attracted the press in the last month and a half that I've been fortunate to represent a team of people. I tend to be the spokesperson, and so I'm the one that gets called up to go on Channel 5 or Channel 15 or wherever it is and talk about the science. I want to talk to you about why there has been so much interest. Essentially for me, it started on Sunday, August 3rd, a little over a month ago. I got an email from a guy by the name of Larry Zeitlin. We've been working together now for about 12 years. He's the president of a little company called MAP MAP Biopharmaceuticals. He and Kevin Whaley, who's the CEO of this little company, have been adjunct faculty with us at ASU in the Biodesign Institute now since about 2002. And so we've been collaborators for a long time. This is a little company, 7 employees down in San Diego. Larry and Kevin both were at Johns Hopkins and just sort of, they got tired of academic life. They got tired of the bureaucracy. They got tired of all the things, and they just pulled up one day and left and moved, got a house along the beach in San Diego, and started writing grants to the U.S. government for projects that really interested them. And it was back in that period around 2000, 2002 that we started communicating, at meetings, and these guys and our team had a very strong interest in trying to come up with new manufacturing technology for pharmaceuticals in the developing world. We had this driving urge. We had to find new ways to make drugs more rapidly available and less expensive. It's really culminated, and then we got this email from Larry, a respondent. He said, "Did you see the news?" I didn't know what he was talking about. I got on the computer and did a Google search for MAP, and MAP quickly led me to Ebola, and then the first press coverage was Bloomberg News, and they reported that an Ebola drug made from tobacco saves the life of 2 U.S. aid workers. Now, if you haven't been the last month, you guys probably been having a good time as you're getting ready to come back to school, so let me remind you what's been happening in the press. These 2 missionaries, both medical experts, were in West Africa. They were involved in volunteer work with this Ebola epidemic that's sweeping West Africa, parts of West Africa. I'll come back to that in a little bit. Anyway, as part of their volunteer work, they came down with Ebola. Both of them have said subsequently that they were planning their funerals. When you get Ebola, you got greater than 50% chance of death. And especially with the current strain that's circulating in West Africa is a strain of Zaire Ebola. It's a genetic variant. It's very lethal if you get it. Anyway, they were on their way out, essentially. And fortunately for them, and I'm going to go through some of the details in this, a shipment of this experimental drug that we've been working on now for over 10 years was diverted from some animal research. It was supposed to go into some monkey trials. It was diverted, sent to Africa. These are the first opportunity to use it was with a Liberian doctor, an African doctor, and he was being advised by Doctors Without Frontiers, the French group, and they recommended he not take it because it was an untested drug. So they sort of made the drug, and I honestly don't know who made these decisions to make it available. It is somebody high up in the U.S. government, and I don't know if we'll ever find out. Anyway, these 2 people were there, and a few fortunate things happened. First of all, it was an unapproved drug. It was sent over there, and fortunately these 2 people were well enough educated in medicine that they could give their informed consent to take this drug. It's totally unethical to force someone to take a drug that's not been tested and proven. So they gave informed consent. second sort of good thing about this was there were still, the progression of disease gets worse and worse, and near the end it goes very rapidly, and you lose all consciousness and everything else. You get organ failure, etc. They weren't at that stage yet, so they could make informed consent, and also I'll talk to you about the animal trials in a bit, they were early enough post-infection that the viral load circulating in their body was still able to be controlled by the drug. And so they were given it, next slide. So this is the MD that was over there, Kent Brantley. He received this experimental drug called ZMapp. This is an antibody mixture. Within an hour, he was reported to show improvement. Phenomenal. No one who gets to this stage of Ebola sickness recovers quickly. He was able to walk out of that healthcare facility either, I don't know if it was the next morning or the next, or the subsequent day. He was able to walk out. They sent a special plane over for the two Americans from the U.S. They took him back and quarantined to Emory University. Less than 3 weeks later, he left the hospital, and the lady left the hospital shortly after that. And Time Magazine has done a story that he interviewed him, and he says, at some point I was told about an experimental drug. It had worked on monkeys but never been tested in humans, and he gave us informed consent. And he said in the story, the doctor decided to give me the drug, and within an hour my body stabilized a bit. Remarkable story of efficacy of a drug. Now, oops, sorry. Within a few hours of these press reports, the conspiracy theories started rolling around in the press, as they were wont to do, and they kept referring to this as a secret drug developed in collaboration with the U.S. Army. You know, something very suspicious about all this. I'm going to give you the background of this. So there's a grain of truth in this because the Army funded it, and it was secret to the press because they don't bother go out and looking at the scientific literature, so they didn't understand all the background research that had gone into it. So for them it was a secret. But anyway, that developed into a lot of conspiracy stories, etc. Questions were flying through the media. Why is it that they didn't give it to Africans? Why do they only give the drug to Americans? And is there more of this drug? I was on a radio show, on a Spanish language radio out of Miami, Florida, about 2 weeks ago, and was peppered with questions. Why are you hiding this drug? Why don't you make the drug available to the rest of the world, etc., etc. It was conspiracy theories that either the U.S. Army or the U.S. government or both are somehow conspiring to let people die and not make it available. And again, I'm trying to give you the background about that. Now, Mary Paul Keeney at WHO helped to reduce these rumors. She came out and endorsed the use of, first of all, of an untested drug under these extraordinary circumstances, and gave a bunch of other things. By the way, I'm going to just mention here, I've known Mary Paul Keeney for about 20 years. Her expertise is not Ebola. She has organized 2 meetings in Geneva on rabies that I've attended because we've been interested in using our technologies for rabies therapy in the developing world. If any of you get bit by a bat today or tonight, you would be taken to your local emergency room, and if they suspect rabies, you would immediately get about 5 cc's of RIG, rabies immunoglobulin, or antibodies. It's purified blood serum from human volunteers who have been immunized or vaccinated with a rabies vaccine. Now, actually, they're usually veterinarians because they have to, for their trade, they have to be vaccinated against rabies. They get extra doses of the vaccine, so they're hyperimmune. They have very high levels of antibody in their blood serum. They sell their blood serum, essentially. It goes up in every emergency room around the U.S., around the developed part of the world, has this in stock. So you'd get 5 cc's shot in the butt, you'd get the antibodies. That would be your passive protection. The antibodies would be taken up, get into your bloodstream, and protect you from rabies. In addition, you usually on your torso you get 6 injections of the rabies vaccine. That's for longer term protection. The antibodies are for short term, in hours to a couple days, a few days, and the vaccine to give you protection longer term. We haven't lost a person to rabies in the United States for over 40 years because this combined therapy works. Mary Paul Keeney knows this very well because she's been trying to implement this rabies protection strategy in the developing world around the globe. But when she saw what's happening with Ebola, another lethal disease, no more lethal than rabies, and about the same time course of infectivity and disease progression as rabies. The two are about the same. So Mary Paul Keeney, very clever lady, says there's some synergy here. And that's why she jumped in and took over this whole program on Ebola protection within WHO. And I'll come back, if you think of it, that might be a good set of questions to follow up on later. Now, so let's jump back to this August, whatever the day it was, I said that Larry Zeitlin called me, or sent an email. I sent emails back to him and I said, well, first of all, you're going to get a Nobel Prize, Larry. And he scoffed, of course, as he does. He's a sort of a beach bum and doesn't take most things seriously, although he is, I would have to say, he's been the leader, the champion all the way through for the last decade on making an Ebola drug. He said, we have sent to Africa essentially all the antibodies we have, this cocktail called ZMapp. For Zaire strain of Ebola, and MAP is the company. He didn't tell me exactly how much they had, but judging by the fact that they've now used the drug on 7 individuals, and the dosage is about 10 grams of antibody IV over 3 doses. So they're getting, if any of you know anything about antibody therapy, this is just a whopping amount of antibodies. It just would swamp your blood serum with antibodies against Ebola. Yeah.
Do you recall the viral load similarities in the patients?
No, unfortunately, she's asking about the viral loads, comparing the patients. Unfortunately, we'll never know, because I'll show you some pictures from where they're coming out of West Africa right now.
Great.
The healthcare, the infrastructure just would not support those sorts of questions. It started with the 2 American missionaries that I've mentioned. They both recovered. This Spanish priest, and probably as old as I am, an old codger, came down with Ebola. They loaded him into this isolation chamber, and you can see anybody getting near him is in these protective suits so they don't pick up the virus. He got 1 shot before he left Africa. And I don't know if it wasn't available or if he was already in organ failure when he got back to Spain, but he died. There were 3 African health workers that got treated after this, and a British male nurse that were treated. One of the 3 health workers died, the others survived, and essentially the correlation was younger people tend to survive, their immune system can come back better, and those who had better access to supportive care. So the ones that got, like the guy in Britain got back to Britain, the Americans got flown back to the US, they're in very supportive healthcare systems. And the other point was all the survivors were relatively early in the progression of disease. They hadn't yet reached the stage of going into organ failure. They were severely sick, but hadn't gotten to that point. And they survived. Now, I will immediately say what NIH says, what MAP says, etc. This does not prove that ZMapp saved those people. We don't have control experiments. We don't have people getting the same type of antibody that's not specific for Ebola. But we haven't done the control experiments. So I'm going to come back and touch on this point repeatedly through here. This is the strangest use of a drug that I have ever seen, because it wasn't approved by anybody. I don't know how the hell it ever got that. Doctors, the drug, and the Spanish priest, etc. So there's a more than a little anecdotal data now, but the question is, what are we going to do next? So this current crisis has been going since about March. At least that's when WHO can track it back. It may have started a little bit earlier, but as of yesterday, when I did the search, there's 3,600 people in Guinea, Liberia, Nigeria, Senegal, and Sierra Leone that have contracted Ebola. I heard on the radio as I was driving in this morning, there's now more than 2,000 deaths, and half of them in the last 3 weeks. So this is a epidemic that's not waning at all. It's taken off like crazy. So the leading question is there more ZMapp? Who's going to get it? When are we going to have it available, etc.? Before I tell you about that, I want to try to explain why I'm involved and why ASU gets some credit for this whole thing. I don't want to say we get the credit. If you get about 100 scientists involved in a room and you ask them to hold up their hand if they participated in making ZMapp possible, there's at least 100 people that could easily claim credit for that. And we've got some of those people here at ASU. Well, I should say, from my standpoint, I started back in 1992 making transgenic plants, that is, inserting a gene into a plant, getting stable localization in the nucleus so that gene expresses to make a pharmaceutical protein. The first one we focused on is the surface protein of the hepatitis B virus. That is the active component in the currently available hepatitis B vaccine. That protein, for all of you who've gotten this, and I hope everybody in this room has had their Hep B shot, I expect you have, that protein came out of yeast because that's the way Merck started making it, and Merck has shared their general technology for transgenic yeast expression in fermentation vats. The vaccine, the hepatitis B vaccine, the cost of it has gone from about $5 a dose down to about 25 to 50 cents a dose, depending on where you are in the world. Now, 25 cents may sound really cheap for a vaccine, but if you're dealing with a third of the global population that's making less than $2 a day in income, 25 cents is significant. And it isn't 25 cents for the vaccine because the government has to provide healthcare workers. You need to make sure that you have a cold chain, that is refrigeration from point of manufacture to delivery. The cost, in general, WHO estimates that the cost of the hepatitis B vaccine is about $3 per dose delivered. And it isn't going to get much cheaper than that if you have an injectable refrigerated vaccine. So back in 1992, I had this academic, naive goal of shortcutting all this and making a cheaper vaccine. Back in those days, we were very well aware that in the last century, pharmaceutical manufacture, particularly for protein drugs, just took off like crazy. We started using mammalian cells like CHO cells, Chinese hamster ovary cells grown in culture. CHO cells are the workhorse for making monoclonal antibodies for all the big pharmaceutical companies. And why CHO cells? Just happened to be that there was a line of hamster ovary cells back in the late 80s, and when you put them in a fermenter, they worked well and they would crank out antibodies. It was just historical accident, but, and I'll come back to this later, but once a pharmaceutical company gets going with the technology, they're very hard to shift because they get all the expertise in-house that they need to work with it. So CHO cells and of course polio has been largely, not entirely, eradicated because we have monkey cells in culture and you use the monkey cells to grow the polio virus. Eggs are used to make the flu vaccine. A variety of recombinant organisms, I have already mentioned hepatitis B, produced in genetically modified yeast. But as all these improvements came in the last century, cost of protein drugs actually increased because the investments got higher and higher. First of all, the capital costs, which I'm going to touch on, it means building your manufacturing facility are going up, and the regulatory costs have increased exponentially. As a society, Americans and Western Europeans in particular have become entirely risk avoidance populations. They won't take any risk. A drug has to be perfect with no side effects, it has to always work every time, etc. That's what we want. If it doesn't, we want more regulations to make sure it's tested before we get those drugs. I'm not saying it's good or bad. That's just life in America, folks. That is driving a huge part of our drug development costs. The other part is what I mentioned is capital costs. If you are going to discover a new drug and take it through the normal FDA-regulated developmental schedule, it's going to take you 12 to 15 years. If this is a new cancer drug, as long as it's a sort of a look-alike to all the monoclonal antibodies that are used today as cancer drugs, like Rituxan, Herceptin, Avastin, etc. All the stins are all monoclonal antibodies produced by Genentech and Merck and Glaxo and all these companies. All of them use the same strategy for production. They build very large facilities to manufacture their drugs. If any of you have ever driven from San Francisco up to Sacramento, you're driving along and there's little Vacaville, a little town off to your left right after you get by Davis. You look at this enormous building, footprint larger than a football field, no windows. That's Genentech's cancer production facility, and I think that's where they make Rituxan. One monoclonal antibody per building, and they had to get the whole building approved by FDA before they got there. They spent about $1 billion building that building. So companies are taking this big risk all the way through here that the drug may fail, and about 5 years before they're actually going to sell it to people, they have to build their facility. If you're an economist and you could back this down a little bit and not take 5 years to get a facility going, you have what's called capital cost avoidance. You're backing off the time when you have to make your investment. You're farther down the developmental pipeline, so you've done more preclinical and maybe even clinical work. And if you can delay your time to build your building, that's money in the bank. You don't have to go to your banker to get that money until you think the drug is in pretty good shape. Now, we recognized that and said, the paradigm's wrong. Get rid of these big damn buildings. Let's find a simpler way to delay the commitment in the scale-up costs. Let's find something else to do. And so, just sort of, this is our thought process. This is back in the 1990s. Infectious diseases are the world's biggest killer of children, etc. And so we had a simple question. Could we change the paradigm? Can we find a different manufacturing platform and make it work? And we did a number of studies, most of which were funded by the National Institute of Health, a few other philanthropic organizations. And the central sort of theme and plan that we did was choose a plant which you can put genes into, and we're only doing transgenics, genetically modified plants, for the first 10 years we did this sort of stuff. And then we chose plants like potatoes, which you can eat without cooking, because all the protein drugs we're talking about would be destroyed by heating. So here's the case. We're just, the preclinical work was with mice. You have them eat one of these genetically modified potatoes that are producing a vaccine or an antibody or whatever it is. Once you had some preclinical data, we would go into FDA. FDA accepted the idea that this pharmaceutical that was in the food is just a food additive. They have rules to regulate that. We got our approval in about 6 months. We went to the clinic, and that's one of our volunteers who volunteered to be immunized in this particular case against hepatitis B, using potatoes and a vaccine for hepatitis B in the transgenic potatoes. To make this odds of this working better for us, we only took volunteers who'd had their hepatitis B vaccine within the last 15 years. Now, all of you most likely have had your vaccine. Right after being immunized with Hep B, if you take a blood serum sample, you can measure the milli-international units of anti-hepatitis B antibody. In other words, there's a very specific test in all your hospital clinics that say, yeah, you have been exposed to the surface protein of the virus. Now, hopefully you got that exposure in the vaccine and not in the disease itself. Now, the test can't tell which one it is, but we knew they had, from their records and their history, none of them had hepatitis B. So, again, all of you have had this shot, you spike up, you get high levels of antibody in your blood serum, and then it decays down. The immune memory is slowed. None of us bother making all this antibody all the time. We only have the ability to recall the production of the antibody if exposed to the disease. And that works beautiful. So in general, we had volunteers whose antibody titers were down around 30 to 50 milli-international units. We then fed them grocery store potatoes in 3 doses, raw potatoes. The only complaint we had was we should have included salt in the protocol because they were starchy. Yeah. But when we gave them our genetically modified potatoes, after 3 doses, they went up to the level that would be equivalent to what they would have had when they first got their injected immunization with a yeast-based vaccine. So this was a proof of process concept, proof of concept set of experiments. And we've done it now with 3 different potential candidate vaccines. But we never really called these vaccines, although maybe we got quoted that way in the press, because I never intended this to be that kids out there are going to eat raw potatoes or anything like that. What we really expected was we would be able to develop simple processing. For instance, take a tomato, freeze dry it, grind it up, and develop a powder which could be put in a capsule and give a capsule as an immunization strategy. And my naive expectation was the brilliance of this, my inherent brilliance would cast a light out there and some company in the developing world would pick this up and make inexpensive vaccines. Well, either I'm not brilliant, which I refuse to accept, or the companies out there had other things to do. So anyway, we've never gotten human oral plant-based vaccines accepted anywhere in the world thus far, although I'm not giving up. But we have gotten animal vaccines. This is a licensed poultry vaccine for Newcastle disease produced in tobacco, and we licensed the technology to Dow, and they got the license for the vaccine. So, and I am hopeful of this technology working. There's groups working on it right now for aquaculture, for immunization of fish, and a variety of other things. But in all this, we never were able to convince any of Big Pharma. And I visited Merck and Santa Fe Pasteur and Glaxo, and I even visited the Serum Institute in India, which is a big manufacturer of vaccines, etc. Nobody really cares. This concept just has got no interest. I can give you 3 simple ideas, but maybe it's return on investment that's more important. But there's no experience in plant biotechnology in any big pharma. There also has been a constant concern about genetically modified foods, especially in Europe. The Europeans still argue over whether or not GMOs are safe. There's an argument in this country as well, but that just clouds the picture. And then Big Pharma really didn't understand how FDA and the regulatory agencies would look at that. I was in a meeting presenting this at one meeting, and George Cyber stood up. He's the Vice President for Research at Merck, and he essentially said, you know, you're just naive, dude. This is, if we're going to be successful, we're sticking with proven technology. So, you know, I must say, it frustrates me to no end that we've spent 20 years on this strategy to make inexpensive oral vaccines, and it's going nowhere at the present time. So, the current business model is financially successful. If you're in big pharma, you got enough risks to deal with, so why change your manufacturing? Okay, everything I did in that period of time from around 2000 up on, I forgot that date. I'm getting terrible. I can't even remember when I did this stuff. Up into the early 2000s, we only put genes into plants to make them transgenic. Then finally, the light hit us, mainly because of a very clever group that we collaborated with in Germany, started a little company in Halle, the old East Germany, now West Germany, a guy by the name of Yuri Gliba, who was a plant virologist. And he was interested in what we did, and we were interested in his knowledge of plant virology, and we teamed up together and started working with things like tobacco mosaic virus. By 2006, after about 4 years of work, Yuri Gliba and his colleagues in Icon Genetics published a paper in PNAS that said they can make monoclonal antibodies in tobacco plants at high yield. That was the first time anybody really got decent expression levels and full-size antibodies. A lot of people have gotten antibody production, but if you get truncated heavy chain or disparate coordination between light chain, heavy chain, you get bad assembly, and FDA will never accept these things. Anyway, he got the right thing. We were developing our own vector system at ASU. Took us a few years longer to publish this one, but we've also produced monoclonal antibodies with our own technology and have patented a vector system, and etc. Incidentally, this one, although it doesn't say it in the title, was to produce one of the monoclonal antibodies that's in the ZMAP cocktail that was used in Africa, and Larry Zeitman and Kevin Whaley, come on baby, Larry and Kevin of MAP were our co-authors on this. They did all the antibody side of this work. So by mid-2000s, we knew that tobacco plants could make good antibody. Now, so let me point out to you how and why. I think why is probably more important. But we take any plant virus. We've got, for tobacco, one reason we work with tobacco is it grows very rapidly, has huge leaves, a lot of biomass. And fortunately for us, it's got a whole bunch of viruses which attack it. And largely because the tobacco industry has been very well funded. People make a lot of money on tobacco for the traditional smoking purposes. They've put a lot of money into disease control, and so plant pathologists across the country have worked on tobacco for a long time. So we know a lot about tobacco viruses. And everything that I'm going to tell you from here on out is really about plant virology for the expression side of things. So you take an RNA virus, and almost all the plant viruses we're interested in are RNA viruses, so you got to convert the genome into DNA, and now you got a piece of DNA, and so usually what you do is cut out some of the proteins that the virus normally makes, such as the proteins that cause the virus to move cell to cell or move within the plant. We don't need that. Or we cut out the protein, the gene that encodes the surface protein of the virus, because we don't want it to make any more virus. So we just re-engineer the viral genome and then put in the genes we want, such as a gene for two genes for heavy and light chain or monoclonal antibody. Put them in the lab, and we're testing our constructs or our plasmids or DNA. You simply take a syringe, no needle on it, put it up on the bottom of a tobacco leaf, The inside of a tobacco leaf is largely filled with air, so you can just squirt a solution in there. And for those of you who would care about it, we're actually putting the DNA into a bacterium, agrobacterium, which is really the shuttle to sort of protect the DNA while it's going in, and then the actual injection into each cell is by the bacterium. That's sort of an irrelevancy. The key point is we've engineered a new virus that makes the proteins we want. We get it into the leaf, we put the plant back in the greenhouse for anywhere from 4 to 10 or 11 days, and then grind it up and go through the extraction process and end up with a clear water-like solution that contains just the proteins we want. So I have sort of gone back on my naivete. I thought we wanted to make a very easy, cheap way of reducing plant material into the drug for oral delivery. No one cares about it. Get totally ignored when I go to meetings, so we switched. We're gonna be just like Big Pharma. We're gonna grind it up and we're gonna purify stuff. Now, what it does is add some expense back in. I'll come back and talk about that. But maybe, well, especially with ZMAT, that's what the vials look like that went off to Africa. It's working. Now, we've also worked with companies to scale this up. In particular, ASU has had a collaboration now for about 8 years with a little company up in Owensboro, Kentucky. There was a big tobacco research facility in this little town of Owensboro, and I can't remember if it was Reynolds or whichever tobacco company it was, pulled out and just left this big facility sitting out there. They wanted to keep some employment in town, so the local hospital bought up the facility and said, what the devil can we do with this facility? It's set up for growing tobacco plants and doing research on tobacco. And we were able to collaborate and get some of our colleagues together, jointly wrote grants together to fund them so that they could develop manufacturing expertise for things we cared about. So again, monoclonal antibodies, you take genes for the heavy and the light chain, put them in so we provided them with our viral vectors. We call it our molecular toolbox for plants. We got bits of virus here and bits of something over here, and regulatory factors, and we put them in, take them out of the toolbox, make something good out of it, and then we can infect plants. These are pictures from 3 years ago. They're more sophisticated today, but the whole idea was tobacco plants are grown in these plastic pans, essentially. There's inert support in there. It's all hydroponics. There's no soil. They're grown in totally closed greenhouses to keep out any disease agents, any insects, anything else. And the next generation of these that I'm going to describe in a few minutes, they don't even use greenhouses now. It's on racks in essentially an old Walmart warehouse. They're using LEDs for the lighting system, but it's essentially it is large production facilities of plants. Plants are moved around on roller belts that are picked up by a robot, dipped into this solution, so you get the modified virus into the leaves of the plant. They go back into another growth room. Oops, sorry. Then after the requisite number of days, before the plants begin dying, they're taken out, you grind them up, and from here on out, the purification protocol is essentially identical to what would be used by Merck if they were growing things in CHO cells. So once you break a CHO cell open and release the antibodies, you're in good manufacturing practice. All has to be under sterile conditions. Same thing here. Once we break open the tobacco leaves and get out, we just say the green juice, you're in sterile conditions after that. And then the protocols are pretty much identical for mammalian cell or plant cell-based monoclonal antibodies. Okay, let's go back to this question. I keep coming back to ASU. This is, after all, we're here at ASU, so we're pumping ASU's story in this whole thing. So how did we get involved? Well, this is a report I wrote, or I didn't write it. The authors are me, Hugh Mason, professor here in the School of Life Sciences, another professor who's left, a project manager we had, and Larry Zeitlin from MAP. So we were writing our report in June 2005. This was at the end of a 3-year study from 2002 to 2005, collaboration between ASU and MAP. It was funded by the U.S. Army Medical Research and Material Command. I'll come back to that. Why the Army in a little bit later. In the end of this report, the last line said, we have demonstrated that Ebola-specific humanized monoclonal antibodies can be produced using plant biotechnology. Essentially the process I just described to you. Humanized is important here. The mice that, the genes for the monoclonal antibodies that we were using came from mice. Now, they came from one of our colleagues in Fort Detrick. Fort Detrick is this big facility in Maryland where they have BSL-4 facilities. That's where you take all the dangerous potential pathogens that you're going to bring in. And it's one of only 2 places in the U.S. where a live Ebola virus is allowed. So they were infecting mice. Mice aren't a very good host for Ebola, but they would get some infection, and the mice would develop antibodies. From those mice, we got genes or DNA sequences for the murine antibodies. Those had to be humanized. There's a whole bunch of stuff in antibodies that I would embarrass myself if I tried to explain them all to you, because I'm really not an antibody nut. But you got to take out T-cell recognition sequences that change the amino acids. Humans and mice recognize antibodies differently, etc. So it had to be humanized, and we did that. I say we in this royal sense. It was actually Larry did all the work. He's the antibody guru and knows how to do this stuff, and he told me what to write in the report. So back to, now let's jump ahead. So that was 2005. We continued our collaboration. By 2011, we published back-to-back papers in PNAS in 2011. One was enhanced potency of fucose-free monoclonal antibody being developed as an Ebola, This is one of the components of ZMAP that's being used today. We had gone off using the same technology. We were making a vaccine for Ebola. Frankly, these guys at MAP have moved a lot faster on their antibody than we've moved in our vaccine, partially because we haven't yet convinced NIH that our approach for a vaccine is better than what NIH is doing, but that's irrelevant. Again, people don't recognize my brilliance. I just don't understand this. But anyway, we have a candidate. In 2011, we had candidate vaccines and monoclonal antibodies. Now, you may have noticed in that last slide that the paper by MAP said a fucose-free monoclonal. Now, I don't know how far you've gone in your biochemistry or immunology to recognize this, but I'll keep it as simple as I can here. Virtually all proteins that are made via the process of transferring the Golgi in a cell, either a plant cell or mammalian cell or whatever, get decorated with sugars, so they become glycoproteins. Almost all the proteins on the outside of our cells, or all the proteins on the outside of a virus, for example, are glycoproteins, meaning they have sugars on them. It's very important in the case of antibodies, and all these little fine details in here are important, but for the start of a complement cascade, which is the way, very important for the body clearing a virus after an antibody binds to it. This glycosylation pattern is very important. And so, what we're going to do, ZMAP is a cocktail, 3 separate monoclonal antibodies. Those antibodies have to bind to the Ebola virus, and when they bind, there's slight conformational change in the virus, in the antibody. Essentially says to our immune system, okay, that antibody has glommed onto something. It's now exposing a part of the sugar residues on here, and it leads to this complement cascade, which leads to the clearance of the virus and the attached antibodies from the body. Now, so I already mentioned to you that we did some humanizing of the gene because we had a mouse gene, had to change it into essentially what a human gene would look like for that antibody. That's all DNA related and relates to the primary amino acid sequence of the two proteins. But if you're going to do engineering on the glycosylation site, that's done by the host that's producing the protein. It's not encoded in the primary sequence at all. So what it meant was to get a fucose-free antibody, we had to work with another group. And this one, absolutely delightful lady by the name of Dr. Steinkellner in Vienna, Austria. Wonderful place to have to go to visit and eat strudel and drink wonderful white wines. Anyway, we buck up and we do all this for science and for collaboration. Anyway, she published back in 2008 the start of a series of papers she's done now on host plant engineering, where she is now genetically modifying the tobacco plant by knocking out certain genes, and in particular genes that transfer either xylose or fucose to proteins, as plants are known to do. So let me just give, this is now out of Zeitlin's paper, this one we've just been talking about, the fucose-free paper. Rituxan, that's Genentech's cancer drug. It's a monoclonal antibody. It's produced in CHO cells. And when you take that antibody out, use very sophisticated carbohydrate chemistry, you can see what sugar groups are attached to the monoclonal antibody that's in the commercial preparation of Rituxan. And it turns out it's not pure, but roughly half of it has this particular pattern of glycosylation. You can see mannose, galactose, glucanac, etc. Terminal glucanac in that particular case. Got some that's got a galactose on it, etc. So that's what a known drug, a $2 billion a year, maybe $3 now, drug looks like. My point here is, when you make an antibody with any living cell system like CHO cells, you don't get out one product. You get out a product that has a degree of variation. And if FDA is looking at this to get approval, they essentially demand this sort of data, and then every time you make a production run, you have to be within about 10% of that. So this is regulation on product by process. It isn't, if you were to make a new aspirin, that's a chemically defined molecule, you got to have 100% of an exact chemistry. If you're making a new cancer drug, you just have to be able to produce something that is consistent. And you have to make sure you're using the same stuff all the way through in your clinical trials. Okay, now when we took, and here's the Ebola monoclonal antibody produced in CHO cells. And not surprisingly, it's got sort of the same thing as what cancer drugs have, but a little bit more variability. But then we took the same antibody, 13F6, and produced it in a plant that Dr. Steinkeller had genetically modified so it can't put fucose or xylose onto its proteins. And we ended up with 90% of this particular species here, where glucanac is the terminal sugar. That happens to be a very common sugar in human cells. Our human cells make a whole variety of things, but this is human-like. And I'm going to come back in just a moment. I hope you sort of remember, this is the one, the delta xylose fucose, the knockout. And this is one where you just took out the glycosylation site altogether, so you don't get any sugar at all added, so it's an aglycone. Now when you they then took and injected them into mice which had been infected with Ebola. And they did it at a very, very low level of antibody. Normally you'd be using 10, 100 times more antibody to get protection. But they wanted to be able to find if there was a little variation in these antibodies. So you do subclinical or suboptimal doses, and it turned out the one that worked the best was this one that came from tobacco plants that had been modified in their glycosylation capacity. In other words, in this particular case, it may not be true for all other drugs, but in this case, anti-Ebola monoclonal antibodies produced in tobacco were better even than the equivalent ones, way better than the equivalent ones produced in CHO cells. So that the conclusion in the publication is that this particular uniform glycosylation pattern is superior to anything else. This is about where the glycosylation data was 2 months ago. This is what we knew about the ZMapp that was shipped off to Africa. So let me just sort of outline where we were here in 2010 yeah, so I'm sort of jumping back and forth in time. Now 2010, we had groups. ASU is sort of the largest group in here, creating this plant molecular toolbox, so we're putting together the ways to make viruses work in plants. We had Dr. Steinkellner in Austria working on engineering the tobacco plant for change glycosylation, and we had MAP in principally working on antibodies. They're the antibody engineers. They're in San Diego doing better things, and all of us caring about tobacco plants. Every group's growing tobacco plants and testing them. But the best we could do in any of our groups was relatively modest amounts of protein production. Remember those big greenhouses I showed you in an earlier picture? We just didn't have that capacity. So all of us were saying, what's missing? We got to get somebody on board who can do scale-up manufacturing. I've already mentioned to you Kentucky Bioprocessing. We were working with them for a number of years, and all of us were delighted when, in 2010, DARPA, which is an arm of the U.S. Army, Defense Applied Research Project Administration, I think it is, DARPA came out with a call for proposals on what they called the Blue Angel Project. I have no idea where that name comes from or what it means, but DARPA does things like that. They come up, Blue Angel Project. And anyway, Colonel Alan McGill is, he's an MD, he was the project manager, he's a colonel in the U.S. Army. They put together a 9-member scientific advisory board, and I was absolutely delighted I got to be on that advisory board. I was the only plant guy on there. All the rest were vaccine experts out of Merck and Glaxo and other places, and I learned a lot from them. Anyway, DARPA said, we want to spend something between $80 and $100 million because we're concerned the U.S. has developed all this basic knowledge in plant-made pharmaceuticals, but if we actually need to use this stuff, there's nobody that's prepared to take us to the dance. We're just sitting here with looking pretty, but not being able to go the next step. This is from their call for proposals in this, back in 2010. They said, we know you can do pathogen identification, say Ebola, but they cared about a whole bunch of things. Target discovery, in the case of Ebola, the surface protein. The candidate therapeutic monoclonal antibodies. So by then we were up in this, and even without the big production facilities, we'd already gotten into preclinical trials, but we're stuck here in large-scale manufacturing. So what happened? DARPA, within 6 months, they had committed $80+ million, and our friends at Kentucky Bioprocessing got about a third of it. They had a press release in 2010. DARPA announced that they're being awarded to get a platform based upon tobacco plants, and the goal of the project is to demonstrate that these systems are scalable for rapid delivery of purified recombinant protein. This is 2010. None of us knew that an Ebola epidemic was going to hit in 2014. But this was more than fortuitous. It just, if it hadn't been for this investment, there would not have been a facility capable of scaling up all our basic research in antibodies to do something useful. But we knew these guys, and we were working on it. So between they finished their facility in KBP about 2011. They immediately started working on a number of projects, one of which was the Ebola antibodies, and we're up to the point that they were making about 50 grams per run on things. And those experimental antibodies were being used in monkey trials to show efficacy. Just last week, a paper came out on the preclinical studies with ZMapp. It's not yet in print, but you can find it on the e-publication in Nature. It was such an exciting paper that Tony Fauci, the director of the National Institute of Health, called a press conference. He had all the major networks in, and he was delighted to say 100% of the monkeys that were challenged with Ebola survived Ebola. They used in these studies 18 monkeys. That may not sound like a lot, but monkeys are very expensive, and the bioethics, it's almost easier to work on people than it is on monkeys. The bioethics of getting approval for monkey trials is important, but it's almost overwhelming. Anyway, all monkeys survived, and the key thing in these studies were that the monkeys could be treated up to 5 days after initial injection with high levels of Ebola. Nobody had ever seen that before. Now, this was published, this data was collected about 8 months ago, just published last week. The batch of ZMapp, which was planned for the follow-up on this, was going to do dose escalation studies in monkeys to find out, do you need 1 gram per dose, do you need 2, 5, 10, normal things that you would do in a drug discovery thing. Never got to that because the next batch of the antibodies that was prepared was shipped to Africa and never went into monkey trials. September 3rd, just a few days ago, Fauci was on the news again, this time saying that Health and Human Services was giving $24.9 million over 18 months to MAP, to make more of the ZMapp monoclonals. In my experience in science, I have never, ever seen money like this come available so fast, so quickly, with so much fanfare from the press. And I'm sure it doesn't, if you've been following this Ebola outbreak, it shouldn't surprise anybody. They have a kicker in there, they could get up to 42 million. I'm sure they got milestones in here, they got to meet batch to batch uniformity studies, etc., a whole bunch of things that are in here. Now, how is it that this is going on? Well, why could NIH ever mobilize this quickly? Because people in the press, I think this is out of the New York Times, but I can't remember for sure, are seeing this every day. This is a dead body in West Africa. Somebody died of Ebola. The major way the disease is spreading is the relatives are coming in, participating in burial ceremonies, washing the body, wrapping the body, and they all get sick. So what's being done now is try to train everybody, and there are crews of individuals who go in with these white suits, rubber gloves, masks, etc., spraying bleach on the bodies, trying to inactivate as much virus as they can. There's a group of the volunteers. I mean, these are heroic young folks. They're putting their life at risk all the time, but they're praying together before they go in to pick up another dead body. Anyway, we're all seeing this in the press right now. It engenders an incredible amount of sympathy and an equally incredible amount of demand for let's do something, let's get out there. So I think that's what Fauci and NIH and everybody was responding to, and money is becoming available. But, and I'm ending with this one, but I just say for you, watch the next 6 months to a year. This is totally outside the ordinary. FDA has a document they call the critical path for drug development. You go from discovery up through here. You do preclinical evaluation. You have at least 2 animal models before you can move forward. You have to do toxicology studies with at least 2 animal species before you can move forward. Then you get into large-scale manufacturing. Then you'll do initial human studies with healthy volunteers. You'll do dose escalation with not looking at efficacy. You'll do all this stuff. That's the 12 to 15 years I've been talking about. And all of a sudden we got coming along with Ebola, and we got to just about halfway through this, and we jumped to humans, way over there. Now we're throwing $25 million into MAP to make more of this. I cannot predict for you what's going to happen next. Are we going to take, if we can show relative uniformity in these batches, are they going to be sent off immediately to West Africa? Who takes the liability insurance? I'm sure that MAP has got some clause, probably 5 pages written in this agreement, that they're not responsible for improper use of anything they produce. Because, anyway, lawyers will come out of the woodwork on this stuff. And who is going to make the decision on whether we should go back to this step, maybe expedite it more rapidly, smaller numbers of animals? I don't know. But I just say, watch this. Next 6 months are going to be really interesting. We may be setting a new paradigm for how we deal with candidate drugs for urgently needed disease prevention or disease treatment. And from my standpoint, since I've been working on plant-made pharmaceuticals, I am delighted with this, with everything about this, because it's validating the use of plants as a production platform that's rapid, scalable, and it's proving to be relatively low cost. Thanks.